Hello and welcome again to another edition of Lost in Science. Half an hour on your radio where we talk about all things sciencey.
I have a joke for you, Stu.
You have a joke for me?
Yeah. What did they give the guy who invented the door knocker?
The Nobel Prize?
That's the one, yes.
Okay.
God.
Yeah, thanks for bringing that up, Chris. But that is what we're actually talking about this week. The Nobel Prizes were awarded in Sweden a couple of weeks ago now, I think. And they actually handed them over. So we were going to look at the sciencey prizes that were awarded, including the Nobel Prize for Physiology and Medicine, which I'll be covering. That was awarded to Yoshinori Osumi for his research into the mechanisms of autophagy.
I see your autophagy, and I raise you topological phase transitions, which was what the achievement that was awarded the physics prize. And I won't list the names of the people now. It's a very complicated one.
I sort of figured out the chemistry one, I tried to. And it was awarded to 3 scientists, and I'll go into that in a bit. But it was for the design and synthesis of molecular machines.
And that sounds cool.
Yeah, it is cool.
So it's a bit more complex than a baking soda and vinegar volcano.
I'm pretty sure that's
thing.
exactly what it
That's
was.
exactly what they did.
Oh,
Yeah,
really? Wow.
yeah.
Wow.
Okay,
That time
cool.
gave Nobel Prize for that.
Well, we'll hear more about these Nobel Prizes in detail later on in the show, so stay tuned. As I said in the intro to the show, I'm going to be talking about the Nobel Prize for Physiology or Medicine. And I guess this one's kind of a bit of both. But this year it went to Yoshinori Osumi for his research into the mechanisms of autophagy. So what the heck does that mean? And why is it worthy of a Nobel Prize, what he was doing? So first of all, autophagy. What is autophagy? Autophagy literally means self-eating.
I see.
But we're not talking about zombies or cannibals here.
Or Ouroboros, that snake that eats its own tail.
No, not Ouroboros.
Ouroboros.
But on a very, very much smaller scale, we're talking about stuff that goes on inside living cells. So, obviously we can tell that the cells of all living things, including us humans, are capable of building copies of themselves based on the DNA code in the nucleus of every cell. But there's a lot more in a cell than just a nucleus. There are a bunch of other parts which are referred to as organelles, and they are the things that do most of the work of the cell. And, every cell is actually doing work all the time to keep us alive and keep everything else alive. So organelles are things like mitochondria, which release energy, make our cells have energy to be able to do things. In plants, they have organelles called chloroplasts, which is where photosynthesis takes place, which captures energy from the sun and turns it into food for everyone else. And there's also a whole bunch of other specialized organelles. There's a whole range of them doing all sorts of different things. So in the 1950s, scientists discovered a new kind of organelle, which contained enzymes capable of breaking up other parts of the cell that weren't being used anymore. So they called this organelle a lysosome. So to lyse something is to split it up, to cut it up into pieces. So what they discovered was the mechanism by which the cells could recycle all of that stuff that was inside them. So anything that stopped working or was no longer needed could be broken up into its component parts and reused to build new stuff, and also releases energy as well, which is pretty handy. Otherwise, the cells would just fill up with crap really quickly. So the recycling is really useful ability that cells have. So in the 1960s, it was observed that there were special vesicles that formed inside the cells, which could carry large fragments and even whole organelles to the lysosome.
A vesicle? Vesicle? What is that? What's a vesicle?
It's like a little... It's like the cell membrane sort of buds off and forms a little bubble around something.
Okay.
And that's what a vesicle is. And then the vesicle carries whatever it's got in it and drops it off at the lysosome for recycling. So they actually called these vesicles, they gave them a special name. They were called autophagosomes.
Right. So they're like the garbage collectors of the cell, are they?
Pretty much. Okay. Yeah, that's basically what they're doing, is going around collecting the rubbish, taking it to the recycling centre for recycling. So people were quite excited about this, and cell biologists have been studying it. And then in the 1990s, Yoshinori Osumi began working on autophagy in yeast cells. So yeast cells are very simple, and they're often used as models for other cells because they're easy to study and they're easy to reproduce and all that sort of stuff. So he wasn't actually sure when he started if yeast cells even had a process of autophagy. So he wasn't really sure. So the first thing he had to do was demonstrate whether that actually happened, so then he could go on and do some other studies. So he found some mutant yeast cells that lacked the certain enzymes that break other things down, and he found that his yeast cells lacking these enzymes filled up with autophagosomes,
Right.
the carrying cells, really quickly because he stimulated them to produce these autophagosomes,
Right.
and there was nothing to break them down.
So they just kept reproducing to try and clear the junk, and there was just more junk.
And there was more junk and more junk, and there was no enzymes to break down the
Right.
junk. So he proved that, yes, they did have a process of autophagy. And he also, once he'd figured this out, he was able to find mutant yeast cells which lacked various genes. And so he would study these different strains of mutant yeast cells until he'd figured out the 15 genes that directly relate to the process of autophagy.
My God.
So he identified just by trial and error of checking out which genes were missing and what part of the process got stopped and all that sort of thing. So you're probably saying, well, that's all very interesting and well done, Yoshinori Osumi. But
That's what I was thinking. Was that what you were thinking?
Yeah.
But what use is it? You might think, why does he deserve a Nobel Prize for finding all this stuff out?
That's really cool.
Well, autophagy errors, so errors in the process of autophagy, can cause major problems in pretty much every organism. So just like imagine if there was a garbage strike, how quickly garbage would pile up around the place. Well, this would happen in a cell if there was nothing to take the garbage away and no recycling centre to take it to. So disruption of the autophagy process has been linked to things like Parkinson's disease, type 2 diabetes, and a range of other diseases associated with aging, and also problems associated with some cancers. So when the cell function starts breaking down, these the autophagy process gets disrupted, and the cells find it even harder to function.
Yeah.
Even though they're already battling cancer. And
Well, I mean, cancer is generally like an error in the cell somewhere. And so you can imagine if the mechanism that's supposed to clean up the cell isn't working, then that's a problem.
Well, absolutely. And the other thing that autophagy does in general is it grabs invading bacteria and viruses that pop up in cells and gets rid of them as well. So it's actually part of the immune system as well. So that if you get any disruption in that, your immunity might drop drastically as well. And also because autophagy rapidly provides both energy and building materials, it's also very important in the development of embryos, and it's important in cell differentiation, and it's important in stress responses. So it's basically this, without this process working properly, all of the cell metabolism starts to break down really quickly. So the fact that we understand the genes for it now is pretty amazing.
I guess, yeah, because like cells work by making new proteins and all kinds of stuff. They're constantly making new stuff from their DNA, and that thing has to go somewhere. Once it's done its job, then you've got to get rid of it. Otherwise, you're just going to clog up with all the used parts.
It'll fill up with tools for things that aren't being used anymore. I guess in a sort of roundabout way, he's been awarded the Nobel Prize for Medicine, but it's really a Nobel Prize for recycling.
The Nobel Prize in Physics for 2016 was awarded to 3 people. Now, the way that works often when there is 3 people is actually they split it 2 ways, and then one of those 2 ways gets split 2 ways. So, yeah, this is the way it works. So I think the Nobel Prize is designed to either be given to 1 person or to 2 people or to 3 people, but 2 of them share that half.
1 and 2 halves?
They do break their medal in half and...
Yeah, I think they each get a medal. No, I don't know. I think they each get a medal, but they they would get the prize money would be split accordingly then. I don't know why I got on this little tangent, but yeah, there were 3 people who got it. One half of it went to David J. Thewlis, and the other half was shared between F. Duncan M. Haldane and J. Michael Kosterlitz for, as the citation said, theoretical discoveries of topological phase transitions and topological phases of matter.
Of course.
I don't think I need to say any more than that. But
it sounds pretty obvious to me what you're talking about.
Self-explanatory.
But we have a few minutes left, so I guess I may as well.
You might as well say
something. Yeah. Now, so what this is a field. I will try and explain what all these words mean. But this is the field of condensed matter physics. So condensed matter physics deals with things like solids and liquids, that kind of stuff, where matter is kind of in this sort of more condensed form, I suppose you could say. And you get it covers stuff like the surfaces of these things, which sounds fairly simple, but really isn't. And kind of more sexier sounding stuff like superconductors. Right, so
everyone loves a superconductor.
Yeah, all these properties of these solids and liquids and surfaces and that kind of stuff. All of it is pretty complicated. And look, but I'm going to try and dumb it down a bit. If any condensed matter physicists are out there listening and I get it wrong, please email us at lostinsci@gmail.com or just tweet us or something and tell me that I've got it wrong. But apart from that, we're just going to charge on ahead. Okay, so what these scientists did, as I said, they studied phase transitions using topology. Now, what is a phase transition?
I know this one. It's when matter changes from one state to another. So from like water, from a liquid to a gas, or from a solid to a liquid, or from a gas to a plasma.
Yeah, those kind of things. Yeah, so it's those kind of big changes in the state of it. But when we get into, there are other kind of phases that you can have apart from just the solid and liquid and gas and those sort of things. Can also be between things like a superconducting phase and a non-superconducting phase, those sort of things, where there's a big jump in its properties, a big change in its properties. Now, one of the examples is where there is kind of one of these more subtle phase transitions, and it's been mentioned quite a bit in the writing about this prize, is something called the quantum Hall effect. So this is, just bear with me here, this involves a material that's placed in a magnetic field. The material itself is it's a 2-dimensional material, so it's a really thin sheet of stuff. And you find that when it's in the magnetic field, it conducts electrons. Electrons move along its edges, but in certain channels, and this conductance only happens at discrete values. So it's got a very stable value. It doesn't change when the temperature changes or when you move it around or anything like this. It only changes when you change the magnetic field by a large amount, and when it does, it kind of jumps up by 1 step, or jumps down by 1 step if you drop it down. So it's got these integer values that's controlled by these large changes in the magnetic field.
So but apart from that, it's not a gradual increase as you increase the magnetic field.
Yeah,
it's a sudden jump when it gets to a certain increase.
Yeah, and apart from that, it's very stable. So that's that jump that's the phase transition when it's jumping from one level of conductance to another one. Mm-hmm. Okay, now. Turns out that these phase transitions, and this is the cool bit of this, is they found that it could be described using topology. And topology is the mathematical study of shapes, effectively. So this is where it gets abstract. So the theory they used is this mathematical thing of topology. So now the way it works, the most important thing in topology when you're looking at a shape and a property of a shape is how many holes it has in it. So... and so that's kind of the defining, not the actual contours and kind of stuff, but how many holes. Because we're in topology, things can be mathematically moved around, they can be remodeled and stretched and squeezed. So imagine everything is made out of some sort of clay or something like that, or some sort of plastic material. So something like a plate, which is flat, and a ball, which is a sphere, obviously, are effectively the same shape mathematically because they can be deformed into each other smoothly.
You could squash a ball into a plate shape.
Yeah, quite easily.
Theoretically, inflate a plate into a ball shape.
Or that kind of thing. But you could squish it around. You could do that. You could squish it around. Now, something with a hole in it, though, you can't make a hole in it without tearing it. So something like anything with one hole will be the same thing. So like a donut and a coffee cup is the usual example people give, because a coffee cup has the handle, makes it topologically the same as a donut, which has a hole in the middle. You can't turn the ball into the donut without ripping a hole in the middle. So that's not a smooth change. So they're quite distinct. We count the holes in things. Like a 2-hole thing would be like a pair of glasses with the lenses removed. 3 holes, pretzel maybe, it's got 3 holes in it. Now, how does this relate to our quantum Hall effect? Well, so it's not the material itself that has holes in it in this case. It's a mathematical model extracted from that, where you map out the energy that the electrons can have against their momentum in this kind of multidimensional energy surface. And that energy surface, this graph you draw, theoretically, has holes in it. And the number of holes you find in that energy surface corresponds to the number of these channels that the electrons can go down. So when you're basically changing the magnetic field, you get one of these big jumps, you're going through a phase transition, you're essentially tearing another hole in the surface. And that explains why, there is such a big jump, but also explains why these things are so stable apart from that, because just like any other changes, like your thermal changes and those sort of things, they're just kind of squeezing it around and they're deforming it, but they're not changing its essential topological property, which is the number of holes. So it is a highly theoretical thing. It's very highly mathematical, but it's very interesting. It's led to a whole kind of, it's changed the way people look at these materials, the way people look at these phase transitions, they look at properties of some of these materials, and it's also led to ideas for new materials. They call these topological materials. I'm just going to say some of the applications. They're kind of, we're kind of talking cutting edge stuff. So even some of this work was done a few years ago. People are still developing new materials based on this. There's a thing called topological insulator, which conducts electricity only on its surface. So there's an insulator inside. There are, and those apparently are used in spintronics, which is kind of a weird sort of electronics that uses spins of electrons. It's not important, but apparently people are making hard drives out of this stuff.
Oh, cool.
And generally there are a few other materials like that. And this stability, this fact that these properties of it are stable until you go through one of these phase transitions, means that it could be useful in something like quantum computing, where you're looking at these very finely tuned electronic properties, and they can be very hard to, say, scale up to high temperatures or to a large scale, because they're very fragile to things moving around and shaking, this kind of stuff. But you've got something where its properties are stable due to its topology, then it makes it a lot more better chance for that. So, look, it is a very abstract, theoretical sounding award, but it leads to a whole lot of practical applications, which you probably don't need to worry about too much. But there's sometimes it's these kind of small things that are working in the background that will have big effects down the track and will change your life without you even knowing it.
This year, the Nobel Prize in Chemistry was awarded to 3 people, similar to the Physics Award. The 3 scientists were Jean-Pierre Sauvage of the University in Strasbourg in France, Sir J. Fraser Stoddart from Northwestern University in the States, and to Bernard L. Feringa from the University of Groningen in the Netherlands. And they were awarded the Nobel Prize for the design and synthesis of molecular machines. Basically, what they did was they developed these molecules with controllable movements. So if you added energy to these molecules, it can perform a specified task. The award was given to these 3 scientists because of the different, very vital role they played to the eventual development of these molecular machines. And so their work spans over about 2 decades, but it's actually quite impressive.
Were they actually working together? Were they working on the same project, or they just all contributed to the outcome of the thing? Yeah, so
they didn't actually all work on the same project. They probably know each other now, at least. But what happened was that each of them played some sort of a role, or they did something impressive, and the others built on the previous one's findings. So it started off in 1983, where Jean-Pierre Sauvage, he linked 2 ring-shaped molecules together to form a chain called a catenane. And the interesting thing is that the catenane was a molecule it wasn't bonded by the traditional covalent bonds, but instead it was bonded by a freer mechanical bond, which meant that each of the interlocking rings could actually move independently of one another. So they could move independently and relative to one another.
And these are really small. This is not like some big ring. This is kind of like a little small of, sort of like a dozen or so atoms or something like, is it?
Yeah. So these are really, really tiny rings. They're like little, 5 carbon rings sort of thing. So,
okay. So
really super tiny rings. And so with the interlocking rings, he was able to basically he could make, say, one turn and the other stay stable. So they're not moving with each other. They're not pulling each other. And then in 1991, Fraser Stoddart took the rings to a further step. He developed what's called a rotaxane, and a rotaxane is this molecular ring that's on a really thin molecular axle. And the ring actually moves along the axle. So he took what Sauvage did in 83 and pushed it a step further. And he was able to make these rings basically travel along a molecular axle. So not only do they move independently of one another, but he could control where they were going and push them in one direction along this axle.
Okay, so it's not like necessarily a wheel that's rotating around, but it's going up and down the axle.
Yeah.
Okay.
And then using these rotaxanes, he was able to develop molecular lift, a molecular muscle, and a molecule-based computer chip. So basically, he was using these little rotaxanes to build little machines. But the next guy actually took it a step further. Bernard Feringa, he actually brought it all together. So he used the work of Sauvage and Stoddart to make the first molecular machine, and he took what they did, and actually when his team added the energy, they were making these machines do a very specific task. So the first thing that they did was made a rotor blade that spins continually in the same direction. So the molecule was basically this flat chemical structure, and it looks like 2 rotor blades, and they were joined together by a double bond and a methyl group. So a CH4 group, methyl group, and they added each of these methyl groups to each of the blades, and the methyl groups acted like a ratchet. So when the molecule was exposed to a UV light burst, the blade jumped 180 degrees, and with it, so it jumped 180 degrees around the double bond, and it took the ratchet with it. Can you guys all envision that?
So it was if they kept flashing it would keep spinning around.
Yeah, and so he found out these properties, and he kept blasting it with UV, basically giving it that energy, and it was creating this spin over and over again. And they continued this, and they made the blades rotate around and around. And the interesting thing was that they were able to control the direction. So they were able to assign which way they wanted it to move. And that's basically how it brought together everything to form these molecular machines, because we already knew that we could create these, or I didn't know this, but these people knew that they could create these independently moving molecules. And then they could also make it do things. And then what Faringa's group did was assign the direction and the movement and the type of movement that you could do with it. So since then, his lab's actually been able to create a nanocar.
So a nanocar?
A nanocar. So
what is that?
Like a car-shaped thing made solely of molecules that can move like a car. So it has wheels that rotate and then a body that stays.
So each wheel is like a single molecule?
Yeah. They all work together and then they all rotate. So the 4 wheels rotate in the same direction at the same time to transport the middle bit of the nano car that is stable forward.
Now the trouble they're going to have is making the differential for the back axle, so when it turns, the wheels can turn at different speeds. That'll be the real trick.
How do you steer a nano car? Yeah.
Yeah, we're going to have to worry
very,
about
very tiny
self-driving
steering wheel,
nano
I guess
cars.
if it runs into something, it's not going to cause a lot of damage.
Yeah.
The insurance would be cheap.
They also they were even able to make a glass cylinder rotate. So they were able to use these molecules to rotate a glass cylinder that's about 10,000 times bigger than the little motor.
So they're powerful motors.
Yeah, they're really powerful, very strong. So anyways, together these 3 amazing findings were given credit for the development of a molecular machine. And this has actually been likened to innovations such as the electric motor back in the 1830s. And the officials actually reckon that the molecular machines will most likely be used in the development of new things such as materials, sensors, and energy storage systems.
If we could combine our 3 Nobel Prizes to make, machines
Evil
made
genius.
out of topological materials that then can destroy things in cells, I don't know what we can make.
Evil genius, too much.
Yeah, it sounds good, though. That'll be the next year's Nobel Prize.
Yeah.
That's all we've got time for on this episode of Lost in Science. Thanks for tuning in and joining us. Lost in Science is recorded at the studios of 3CR in Melbourne and broadcast across Australia on the Community Radio Network with the financial assistance of the Community Broadcasting Foundation. If you want to talk to us, talk back to us, you can get in touch. We have a Gmail account, lostinsci@gmail. You can also find us on Twitter and on the Facebook. And if that's not enough Lost in Science for you, you can always tune in again next week, where the team will once again get Lost in Science!
Thanks for listening to a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online.